turnout today for we have a great turnout today for a great speaker. I'm really delighted. It's my honor today to introduce my friend and colleague, Dr. Vadim Gladyshev. Dr. Gladyshev is a professor in the Department of Medicine at the Brigham and Women's and the Dana-Farber Cancer Center and associate member at the Broad Institute. And he's highly regarded for his original and penetrating work in the biology of aging. He's been recognized with top honors. He's a fellow of the AAAS, and in 2021, he was elected to the National Academy of Sciences here in the US. He's recognized not only for his work in aging, but also in nutrition. 2018 was awarded the American Society of Nutrition's Osborne and Mendel Award for outstanding basic research accomplishments in nutrition. Dr. Gladyshev uses a wide variety of approaches, including molecular genetics, systems biology, computational models, comparative biology. And with these approaches, he's made major contributions to the field of aging. He's extremely prolific. His publication record has led to really fundamental insights on interventions to extend lifespan, on the basic mechanisms of aging, on biomarkers of aging. I should mention that he's organized the Biomarkers of Aging Consortium, which is having its meeting here in Boston later this week. And on top of all this, he's also made major contributions in the field of selenoproteins, independent of aging. So we're really fortunate to have him as our colleague here in Boston, and especially to have him join us here today for our weekly seminar at HNRCA. So with that, I will just remind you that to please put your questions throughout in the Q&A section, and after Vadim's seminar, we'll read them out for the Q&A. Over to you, Vadim.
Thank you, Daniel, for the very kind invitation. And for the introduction. And it's really a pleasure to be here presenting in this seminar, because I know many of the colleagues that are part of the center and or gave talks at this seminar series. So it's really a pleasure to do it myself. So let me share a screen and start. So I will tell today a little bit about biomarkers of aging, longevity, and rejuvenation. And this has been, in my mind, a very important development in the field in the last 10 years. And there's a lot of kind of uncertainty, and on the one hand progress, but on the other hand uncertainty in the field. So I will try to present what we're up to in this area. So, but first, it's important to discuss what we are trying to quantify when we say biomarkers of aging, longevity, and rejuvenation. What is actually being, quantified here. And the first one is aging. So important to ask what is aging, because depending on how we define it, then we might develop different types of biomarkers. And defining aging has been very difficult, actually, because we use the word aging to designate different processes. Some people think of aging as the increased chance of dying with age, like, for example, many demographers would use it. And of course, the mortality assays and Gomper's equation have been like major tools in the aging field. But other people say, no, mortality is just one of the manifestations of aging, and the actual aging is maybe a continuation of development or loss of function, or it could be any age-related change. That's, for some people, that's what aging is. And so in that sense, even though these features, they go hand in hand, they are, strictly speaking, not the same. So we end up with no consensus and in fact, again, so thinking very differently about the aging process. And as we think differently, we also would develop different approaches for it, right? So another way to say this is that we could look at aging from the perspective of mortality, but with the understanding that mortality is typically caused by diseases. And then if we, quantify incidence of age-related diseases like Alzheimer's disease, diabetes, heart disease, so on, then we say, well, diseases, are precluded by functional decline. And functional decline happens because of accumulation of damage and other negative consequences of life. So actually, in my lab, we tend to think more at this level with considering the subsequent kind of layers as manifestations of this initial kind of molecular changes. And so when I think about the aging process, I think it's this way. So let's consider an enzyme. The enzyme converts a substrate to a product. And so this function, kind of conversion of the substrate to product, is something that is purposeful. It has a particular purpose. This enzyme is needed to convert a substrate to a product, right? But at the same time, when it catalyzes this reaction, unavoidably, it also produces something else, because enzymes are not perfect. They are composed of only 20 types of amino acids with imperfect, this protein globule. So they are imperfect. So there is non-zero likelihood that the enzyme will produce something else. Maybe it would react with another substrate or another molecule, or will produce a byproduct or something else. So, but this consequence is unavoidable and without any purpose. Enzymes are not evolved to produce this damage. It just happens because they have this blue function. So to me, this aging part, the red part represents aging, and the blue part represents life. And they kind of go hand in hand. In a sense, aging happens because we live. And if we extend this concept from this enzyme, we can say that this damage could be represented by many different features. For example, by products of metabolism, or mutations, OP mutations, and all kinds of other known and unknown forms of damage, which would come from every single process. So basically, this means that every cellular function contributes to this damage accumulation, and none of the damage forms is major. It makes it really difficult to study the aging process because it comes from so many different flavors and layers of biological organization. Also, another feature is that aging to me is a quasi-programmed. It's not programmed because there's no purpose in aging. Like there's no purpose in making this damage, but this damage often is non-random. Also, of course, there is also stochastic damage, but some of it is non-random because it's encoded by a particular enzyme. This particular enzyme will produce a particular type of damage, right? Not just any damage. And therefore, it appears quasi-programmed. That's why we age quite similarly. For example, now the maximum lifespan of human 122, and many people in developed countries live like up to 80s and 90s even, right? Very close to the maximum lifespan. Quite amazing. So in terms of the damage, I view this in a very broad sense. We often use this word deleteriome to encompass all age-related deleterious changes, not just molecular damage itself, and which comes from, again, so consequence of life. So we have this model in mind about the aging process, and then we use it in all subsequent kind of studies in the lab. And so the way the lab is organized is that we study first longevity, and here we study longevity at the level of different cell types. Some cells live short, like cells in the blood, some long, like cardiomyocytes. Longevity across mammals, some mammals live short, like mice, some long, like whales. And also longevity interventions, because we know that many interventions, pharmacological, dietary, genetic, they are known to extend lifespan. So we can study these multiple levels, and this results in what we call signatures of longevity, which are omics patterns indicative of the potential of the system to live long. They may or may not change with age, but they change with the change in longevity. At the same time, we study aging, and here we study changes that happen with age, and we try to quantify them with various biomarkers, like, for example, epigenetic clocks, transcriptomic clocks, organ-specific clocks. They result in actionable biomarkers of aging. And both biomarkers of aging and signatures of longevity are really needed to identify interventions and test them. So for example, in the signatures here, if we find an intervention that changes the system in the direction of that signature, it could be a potential geroprotector. And when we test geroprotector, we need to use aging biomarkers to quantify that the organism ages slowly, slower. A third component of what we study is rejuvenation, and this is something very new in the field, and to me, rejuvenation is a conversion from an older state to a younger state in the system. But it's not necessarily a reversal of aging, because aging is, it just has so many interactions and processes, and all the damage forms have so many different flavors. Some of them are stochastic, like a mutation in one cell, another mutation in another cell, or epimutation or byproduct. It's impossible to go all the way back in the aging process. In that sense, aging is irreversible. But to me, it's still possible to target a particular state of the organism to make it younger, not necessarily in the same process, like not in the opposite direction to the aging process. And this is called rejuvenation. And we also develop signatures of rejuvenation. And then we trying to use them to target aging and rejuvenation, developing new interventions and study them in the lab. So just one example of bot longevity is, it's from a paper we published last year, this is by Alex Tishkovsky. And basically here what we do, we study mammals, like longevity across mammals. We have about 40 species of mammals that we analyze at the level of RNA-seq across different organs and many representative of each species. And what we find is that if, for example, a gene shows increased expression with the increase in longevity across mammals, like in one organ, in the brain, typically it shows the same in other organs. If a gene shows a decreased expression, typically there is a decreased expression in other organs as well. It's very interesting, in a way counterintuitive, actually. Now, we could relate changes according to longevity with changes according to aging. And this is what we've done. So here in blue are changes according to longevity across species, like brain across mammals, kidney across mammals, liver across mammals, and the residuals. And you can see they cluster, so they correlate with one another. So the changes that we observe in the brain correlate with changes observed in the kidney or in the liver across mammals. This part is aging, like changes during aging of different species, like muscle across mammals, brain across mammals. Human means different tissues of human, global means different species and different tissues. They also correlate with one another. But also very interesting that consistent changes happen during the aging process at the omics level. And here is the interventions that are known to extend lifespan. When we combine them, for example, interventions of the like calorie restriction type, like acarbose, canagliflozin, FGF21 overexpression, growth hormone receptor deficiency, they all change actually organisms in a similar way, so they cluster with one another. But what is interesting, when we try to relate these three types of aging longevity to unite them, and we find very interesting observations. First, if we relate longevity across species with aging, you can see they're slightly positively correlated, which is strange. It would mean that longer-lived species that are actually older at the same kind of proportion in their life. They are literally older. Why would they be older? That would be very strange if they are especially longer-lived. So our interpretation is that during the aging process, damage accumulates, but organism responds to damage by activating protective systems. And similar protective systems are used in long-lived species. So in a way, long-lived species are not older, they are just better protected because they have this more advanced adaptive systems. They are involved in damage handling and clearance. So another interesting observation is that there is no correlation between longevity across species with longevity across interventions. And this was really surprising because the idea in the field is that we study long-lived species. We find a process that is responsible for their long life, and we transfer this process to like a short-lived species like mouse. And we should be able to use it to extend mouse lifespan. But we find that the way nature extends lifespan and the way we do this in the lab is very different. So our interpretation is that in the lab, we typically target single genes, or we use single compounds, or maybe we target like a single pathway. So it's a very simple kind of interventions, whereas what nature does, it manipulates many genes and pathways simultaneously. And the combined effect of this change is very different from what we do in the lab. To me, it suggests that the studies of this comparative biology of mammals has a great potential because it would allow us to identify completely new ways to extend lifespan, hopefully in the future. Not something that we find at the level of interventions when we can do it like 10% or 20%, but much more radical changes. Like we observe across mammals, there's 200-fold difference in lifespan, like from like some shrews to some whales. So, but it's very difficult because again, so it would not be, there wouldn't be like a single genes or single compounds or single like lifestyle changes that could recapitulate these natural changes in lifespan. But as long as we know this vector of change, then we could utilize it. Now I'd like to tell you a little bit about biomarkers. And biomarkers, or sometimes called aging clocks, have been started by this area, started by Steve Horvath about 10 years ago. And when he developed his first human multi-tissue clock. Now it's a huge proliferation of studies, and clocks are published literally every day. So it's very difficult to overview this area. I will just tell you how, from our own work, how it works. So this was a paper published already 7 years ago. It's the first mouse epigenetic clock. And this is the actual age of the mice. This is predicted age of the mice. And these are 16 age groups. And you can see there is very good correspondence in the actual age in mice and in the predicted age in mice. And the way we do it is that we sequence the blood of these animals with the method called reduced representation bisulfite sequencing out of millions of CpG sites that change, that detected. We identify some sites that change methylation with age, increase methylation or decrease methylation. And within this group of sites, we identify those which are particularly good at quantifying the aging process. In this model, it was 90 CpG sites. And so when we apply this model to mice that have been subjected to calorie restriction, you can see that at any age that we test, the mice are predicted to be younger. So this means that this model works because we know that calorie restriction extends my lifespan, and when we apply this, we already see that the mice are literally younger when quantified by this clock. And what was really surprising to us is that when we applied this clock based on the blood to reprogramming somatic cells, fibroblasts in this case, to induce pluripotent stem cells, and we find that this were like 10-month-old mice, fibroblasts from the kidney and the lung. And when we reprogram them to iPS cells, we find that the age is predicted to be 0. Of course, these are embryonic cells. We expect them to be very young, but it was surprising that the biomarker developed for mouse blood actually quantifies this conversion to embryonic state as well. So, but the question is, how do we actually develop the best biomarkers? Because the initial biomarkers that have been developed, like the one I showed you, are trained based on chronological age. So we call it chronological clock sometimes. So trained on basically on the progression of time. This direction is shown here. Another way to quantify aging could be we could quantify mortality. And there are clocks like, for example, Grimm age, epigenetic clocks that predict a future mortality, time to death, right? They're trained on that feature. There are clocks that are trained based on incidence of age-related diseases. Some clocks are trained on functional decline, but there are no yet clocks that are trained on damage, but we are working on that as well. But the question is, how should we quantify aging? And I already told you that we use the word aging to designate different processes. And therefore, there would be different clocks that are developed, and they quantify slightly different things as during the aging process. And again, so the kind of chronological time or rate of change or mortality or diseases or damage or something else. And nobody knows what's the best way to do it. So for this, we and others organized a consortium and called Biomarker of Aging Consortium, which brings together leading labs in the field, I don't know, 30, 40 labs or so. And this is the first paper we published as a group about a year ago. And since then, there have been a few other papers published. Actually, this Vadim, who is an instructor in the lab, he really was the first to have this idea and put most effort to organize the consortium. And so he's the really key person here, along with Jesse Paganik, who is also instructor in the lab. And so this is the website, and I would like to mention, as already Daniel mentioned, the conference we'll have next week, actually this week, this Friday already, about biomarkers of aging. Biomarkers of aging, we expect 400 to 500 people to attend this meeting. So related to that, this group set up a challenge, biomarker of aging challenge, because now papers are published, like, thousands of, I don't know, millions of papers, and everybody publishes clocks and they say their clocks is the best, but there's no fair comparison. So what we did, we collected 500 samples from our Mass General biobank, and we know who these people are. We know how long they lived, because the blood was collected way before, like much earlier, and then since then it was like 10 to 15 years past, and so some of these patients died, and we collected epigenetic, transcriptomic, proteomic data for them, and we asked people to participate in a challenge of predicting like chronological age of these samples, future mortality, and so on. And this is how it works. So we have like over 500 submissions to this, and 37 teams competed, and this is the results. They're going to be discussed extensively at this meeting later this week, and the winners will present their work as well. You can see these are the kind of entries, that submissions of new clocks, and these are the existing clocks. You can see the at least mean absolute error is much better in this new submissions. So there's a way to go in improving the clocks, and this is how these top submissions fare in this data set. This data set from 20 to about 100 years, and actual age and predicted age. You can see they work really well in predicting age. But this is the age. And at the meeting, we will also announce winners for the second phase, which is the mortality prediction. That's much more difficult because there are fewer data sets available to train the clock, but still there are many teams competed, and then there will be some winners. And the third phase is going to be in next year on predicted multimorbidity. And there are cash prizes associated with that, and we have collaborators that help us at various aspects, as well as many volunteers. It's really now become a large organization. I will tell you briefly about the advances in our own lab here. This is one paper we published earlier this year by Albert Ing, who is a very talented graduate student, and he developed the first causal clocks. Causality is really critical, because when we train the clock for any age of the change, whether it's like we train based on chronological age or future mortality, or it doesn't matter, we train on kind of damaging changes that happen during aging, but as well as neutral changes, but also adaptive changes, kind of things that respond to this damage, because they all have age-related structure. So he was able to separate damaging changes from adaptive changes, and then he developed clock based on them, calling them age and adapt age. And you can see they all work in predicting the age. But when we apply them to reprogramming cells, they show very opposite direction. The clock based on damaging changes shows rejuvenation, but the clock based on adaptive changes, it's not. And which is very reasonable, I think, because adaptive changes and protective changes, if we destroy them in some particular intervention, we may expect any kind of outcomes, not necessarily rejuvenation. Another challenge in the field is the challenge of stochasticity. And that's another paper in Aging we published recently by Andrei Tarhov and other lab members and collaborators. And he addressed the question of stochasticity. And here is if you have a useful state, if, for example, aging is involve co-regulated changes, we expect like this: as one side changes, typically another side changes very similarly. Or like sometimes stochastic changes, like all the changes might be completely random, or maybe mixed changes. And when he studied, he found many co-regulated change. So if like one CPG side changes, typically there are many sides change very similarly. You can predict one from another. But at the same time, he also found many stochastic changes. So in the end, it appears that aging involves both stochastic and regulated changes. And in the same issue of nature aging, there are two other studies, one from Björn Schumacher's lab, another one from Andrew Tischendorf's. They also kind of found similar, they have similar findings. So I wanted to give you a few examples of how we apply epigenetic clocks. And in this study published last year, this was done by Jesse Paganik, who is an instructor in the lab. And he found that under condition of severe stress, biological age is increased. And once the stress is relieved, biological age goes down. We don't call this process rejuvenation because it's kind of increased under stress and then kind of go into the normal level. So it's age reversal, but not necessarily rejuvenation. And there are four kind of examples that he described: major surgery, severe COVID-19, when somebody is in the hospital at the ventilator, biological age is the highest. Once the person recovers, the biological age goes back to normal. Also pregnancy. During pregnancy, we find that women increase their biological age with the highest in the third trimester, and predicted biological age is decreased postpartum. And also parabiosis. And now we have many other cases. The easy way to think about this is that if we consider aging as the process of damage accumulation, under conditions of severe stress, organism is unable to cope with that damage. Damage accumulates. And once the organism recovers, that damage may be partially or fully cleared, and therefore biological age would be decreased. Another example is from the work we published in Nature Aging last year. This is work of Bahan Zeng, who at the time was a graduate student, now stays as a postdoc in the lab, and collaboration with Jim White. He published the study of heterochronic parabiosis, where we connect mice, young mouse and old mouse, for 3 months so that they develop joint circulation, they exchange the blood. And then after 3 months, we separate them and follow what happens in terms of biological age 2 months later. And also wait longer until they die to assess the effect on lifespan. So on the right, here is the effect on predicted biological age. And you can see, like, for example, comparing this one and this group and this group, that after 3 months, old mice are rejuvenated. And the effect stays even longer. Like after 2 months, they still remain rejuvenated. And this is in the liver, in the blood, and we see this in other organs as well. The mice literally become younger, old mice, after they are connected for 3 months to younger mice. And when we look at the lifespan is also extended. So they live longer. One caveat, I guess, here is that the effect on lifespan is not very strong. I mean, significant, but kind of really minor compared to the effect on rejuvenation early in life. And so our thinking is that when after completion of this procedure at 3 months, and then maybe even later, a little bit later, the old mice, they literally rejuvenated very strongly, but then this effect diminishes over time. So the mice are still observed to be live longer, but not as much as expected based on the initial rejuvenation. So I would like to mention a couple of studies which are unpublished. And the first one is this transcriptomic clock that we developed. This was done by Alex Tishkovsky, and he's an instructor in the lab as well. And he developed this very robust transcriptomic clock in mice, rats, and humans based on 4,500 samples. And this involves many studies where we observe a reduction in lifespan or extension in lifespan. This, for example, we sequenced many samples from the ITP project, as well as many other samples. And so this is how the predicted the clock trained on chronological age works. This is the actual age, this is the predicted age, training set and test set. You can see it works really, really well in predicting age. This is trained on chronological age. But we also trained the clock on future mortality, time to death. And this is also can be developed in a very robust way. This is the true kind of findings, and this is predicted findings in terms of mortality. training set and test set. There is correspondence, really good correspondence. And this is the application of the mortality clock to test or validate interventions that shorten lifespan or extend lifespan. And you can see, for example, these are control mice, and this is the mice subjected to lifespan extending interventions. And you can see that the clock can separate controls from lifespan extending interventions as well as from lifespan shortening interventions. So, of course, this also means that this clock would be applicable to test other interventions in the future, candidate interventions. But the advantage of RNA-seq clock, especially compared to epigenetic clock, is that we quantify changes in genes, and we know what the genes are, we know what the pathways are, so we are able to deconstruct the entire transcriptome into modules, and then develop clocks for chronological age of mortality for different modules, like, for example, module of innate immunity inflammation, or adaptive immunity, or heme metabolism, and so on. And each of them would represent the aging process. So sometimes if we are interested in aging, like accelerated aging, for example, of a system, we want to test whether particular modules contribute to that change. Or the same way for rejuvenation. If you observe a rejuvenation, sometimes we want to see rejuvenation is based on which particular pathways in genes. And so we developed these clocks for each of these modules, and the clocks also, many of the clocks work really well, and we actively apply them. Also, we apply this to diseases, and we find that these are mouse models of disease, all kinds of diseases, Alzheimer's disease model, a chronic kidney disease model, neuropathy, ischemic stroke, and in each case, we observe increased biological age. It's actually quite interesting that the disease tissues, based on this clock, are predicted to be older, almost universally. That I found this very interesting. So another application of these biomarkers is in development, because previously when we ask like scientists in the field, when does aging begin? And most people say, well, at the completion of development, or maybe at a puberty state, or a time minimum mortality. But when we apply these clocks that are trained for adult ages to early in life, we find that they tick, like a tick at 15 years, at 10 years, at 5 years, even at fetal stage, they already tick. So to us, this suggests that the aging begins very early, and we wanted to know when exactly does it begin. And we find that by applying the epigenetic clocks initially, we find that clocks begin to tick at this stage. We call it ground zero, very early in life. In humans, approximately 3 weeks of life, but not very early in life, not at the zygote state, is a fertilized egg. We find that the zygote, the age of the zygote is not zero. It's a little bit above zero. And then the predicted biological age goes down later. Well, still early in development, but maybe 2-3 weeks later. And then from this point, we see that the biological age can be quantified as increasing. This is very interesting. And we don't know yet mechanistically how exactly this rejuvenation occurs, but it seems to be universal, perhaps across all vertebrates. And I would stress that this is, so far, the only natural process of rejuvenation known. It would be very interesting to find out how exactly cells are rejuvenated. And maybe if you understand mechanistically this, maybe apply this in the future to somatic cells. And of course, this is still it's not 100% validated because the studies are only from one lab, from our lab. But these are the studies, this is a conceptual paper, and then we find this in mice. And then also we developed, this Alex Sharp developed a completely different single cell epigenetic clock. It's a probabilistic clock. And when we applied, also works, shows the same. And then we find this in humans, and in frogs, we have a bioarchive, not yet published study. And Csaba Kerepesi and Alex Sharp are key here in this particular project. But now, because we have transcriptomic clock, we wanted to test, okay, maybe this process is just a epigenetic process. How about the transcriptomic rejuvenation? And when we train the transcriptomic clock for late life and apply this to development, we observe the same. Interestingly, the zygote is not zero, the age is not zero, and then we observe this robust rejuvenation to ground zero. this is chronological clock and this is mortality clock. They show the same. So we're very pleased that this additional, completely different method also has the same predictions. Again, so it would be important to validate this finding, by other labs and by other methods, because it's potentially of fundamental importance. This is another example of this. This is a single cell mouse data set from 6.5 post-conception to 8.5 days post-conception. And we observe by applying our transcriptomic clock, there's robust rejuvenation. The predicted age is consistently decreased during this process. And we ask a question, this rejuvenation happens in which cells? Because we observe this in the whole embryo, and with the transcriptomic clock, we now can analyze different cell types at a single cell level. And we observe that all cell types are rejuvenated, except perhaps, well, actually not perhaps, except for extra embryonic tissues. It seems like during development, extra embryonic tissues, for example, cells that would become placenta late in development, they are not rejuvenated. And also it would make sense why they should be completely rejuvenated. I think it would be important to rejuvenate the cells that would in the future would become the embryo itself. So now there's another small story which is also unpublished, and here is kind of relates to organismal versus organ aging. You often ask a question like how old a particular person is, and we say, well, this person may be I don't know, 80-year-old, but looks like 70-year-old or 60-year-old, and we predict the age of the organism. But in fact, different part of the organism may age with slightly different rates. And I already showed you the example of the application of our transcriptomic clock to disease state, and where we find that disease tissue is older, predicted to be older. So what if this is more universal and different organs have slightly different aging states within the organism? How do we even quantify that? It would be very difficult to do it. So one possible solution to this came from this paper. I think it's an important paper published by Tony Viscaray's lab. He's at Stanford last December in Nature, where they find that there are so-called like kidney agers, like 2% of the population, they age more in the kidney, and like 2% population ages more in the heart, and maybe 2% ages like across multiple organs and so on. And they were based on quantifying proteins that are secreted by different organs. And we've done very similar studies, but perhaps at a better population. We use UK Biobank, which is like, there's 10 times more subjects available. And also importantly, there's mortality information. So we are able to develop clocks based on chronological age and also based on future mortality, based on this data set. Again, so what we do, we have access to 53,000 proteomic profiles of UK Biobank subjects, and we also identify proteins in the plasma that come from particular tissue. For example, we look at the gene expression and we find that certain genes are expressed, let's say, in the brain only, and then we later detect them in plasma. We infer that these proteins were made in the brain and secreted uniquely to the plasma, and by quantifying changes like age-related changes in these proteins, we can predict the age of the brain. Similarly, we could predict the age of the liver, of the kidney, of the lung, and so on in the same person, based on this, by quantifying proteins that are uniquely secreted by particular organs. And this slide shows the clocks that we developed, these organ-specific clocks based on the proteomic profiles, and also the relationship between these clocks. So residual, basically it shows residuals. Yeah. So here's the relationship between the shown on the left chronological clock. This is mortality clock. You can see if somebody is older in the brain, typically they are older like in the liver as well, but other organs, whether we consider chronological model or mortality model. So this suggests that there is some kind of coordinated aging across organs. So in general, in the population, if somebody is like old in one organ, they would be old in another organ as well. So this accelerated age in one organ kind of transferred across organs. they assimilate the age of each other. But at the same time, we also know that this process is not perfect. So some organs may age faster or slower compared to other organs. And how do we study that? So what is interesting is that when we take like the lung model, these are the lung proteins that are secreted in the plasma, detected in the plasma. We build a clock based on them, predict the age of the lung, right? We ask a question, which diseases are predicted best by this model? The first disease is COPD. If we take the liver specific clock, the first disease that is predicted is liver cirrhosis. And this is the kind of first generation means chronological model, mortality-based model. Mortality works best for predicting diseases, actually, always better. So now if we try to relate in a more global way organ-specific aging and diseases, and this is shown here, we again find that people with COPD, this is the predicted ages in different organs, like, or age in the artery, in the kidney, intestinal age, and so on. But they are older COPD, older in the lung here. Yeah, maybe also a little bit older in the immune system. People with dementia, they are older in the brain. So there is unequal aging of different organs. So on the one hand, they are in part assimilate age of each other. On the other hand, there's unequal aging of different organs. There's also interesting that different organs and diseases, they cluster. So they so tightly influence each other that it's hard to separate them, at least with the current methods. And these are many metabolic diseases and many organs that like they talk to each other very, 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 very tightly, like the heart and lung and kidney and so on. Now, we could relate this organ aging to many features like phenotypes. For example, we could relate them to professions or lifestyles or diets and so on. And this is one example, is smoking. Smoking, of course, we know it's bad. But is it universally bad for all organs? So we could test this idea using these clocks because it's, again, the same people. We look at the same people. So we could ask a question, how, to what extent different organs are affected. And we find that the strongest effect is actually for the lung. So, which is not surprising, of course, for smoking. But of course, there are also many negative effects observed for other organs as well. But no positive effect of smoking, interestingly. But then we ask a question, what about alcohol consumption? Is it good or bad? And if it's bad, for which organs? And we find that in general, it's bad. For example, especially for the kidney, also intestinal aging. But we were surprised to observe a positive effect for the artery. So it's unexpected, a little bit unexpected that alcohol consumption would actually have a positive effect. So just to extend this kind of observation, here I show you aging based on the clock of the artery. Artery means like proteins are made in the artery and released in the plasma. We detect them in the plasma, but we use quantify age of the artery, right? And we associate this with the effects of different diets, or dietary components. And we find that wine is good, white wine is good for the artery. Yogurt has a slight negative effect. But if now we replace artery clock with the intestinal aging clock, the effect is opposite. Now wine is damaging and yogurt is good. So what does it mean? It means, I think we should really, instead of thinking of kind of universal longevity interventions, or maybe in addition to think of universal longevity interventions, we need to think of personalized longevity interventions. We need to know who can benefit from which intervention. Even though we know the calorie restriction is generally good, not everybody can benefit from calorie restriction. There are some people who age in a particular way in their organs, such that calorie restriction would be damaging. In the same way, it would be like the example here of the arterial and intestinal aging shows that somebody ages more according to intestinal clock. They maybe should eat sugars, yogurt, but don't drink, right? But somebody ages more in the artery, maybe it's okay for them to drink wine, but avoid like sugars and yogurt. So this is really would be different strategies, right? Depends on how particular aging phenotypes across organs are observed for that particular individual. So what I think this study shows is asynchrony in organ aging. And we need to think how to target weak links in organismal aging and really think of personalized longevity interventions in addition to universal. Because these weak links, organs with accelerated aging, these are the organs that would lead to development of the diseases in that organ and eventually would contribute greatly to mortality of the individual. So by targeting these organs with accelerated aging early before they develop disease, I think would be quite beneficial. And there are now ways based on this modeling to identify such potential interventions and then test them. So this is our model. This is the zygote where like initial cellular life begins. And we think that this is the place where aging begins at ground zero, but 3 weeks of life in the human, maybe like a little bit less than 2 weeks in mice, like a week or so. And then different organs and systems age and they talk to each other. They age with somewhat similar rate, but not identical rate. And sometimes some organs age more for whatever reasons. It could be a genetic contribution or lifestyle or just simply kind of random effects. But this is not a simply older state because again, so it's non-autonomous aging. So these organs with accelerated aging, they talk to other organs. In a way, their age is decreased by other organs, right? So it's both older and altered state. And we don't know yet how to describe it, this particular state of unequal aging across tissues. But I think it's kind of important to follow on that. So another question in the field that is being asked often, is aging a disease? There's a lot of push to classify aging as a disease so that it can be treated. I'm not really sure this is the good direction myself. I don't have a strong opinion, but 8 years ago, we published a paper stating that aging is not a disease and actually not a disease. And just the question itself is wrong in my mind. But now with this new understanding, we could reverse the question and ask it in a different way. We ask whether a chronic age-related disease represents a manifestation of aging, whether a disease can be viewed in the aging paradigm itself. And one way to think of that is that disease could be viewed as a systems or subsystems aging. Or maybe diseases can be viewed as pockets of non-autonomous aging, where some parts of the organism, be it a particular system or organ, or maybe a smaller set, maybe even like one cell, could show accelerated aging. And sometimes this accelerated aging, but not simply accelerated, but also altered, that state we call a disease. So I think this has a potential to unite or unify kind of aging and disease paradigms, but not from the perspective of diseases, but from the perspective of aging. So finally, I'd like to summarize what we discussed today in this seminar. I told you a little bit about longevity signatures across cells and species interventions, also difference between species longevity and longevity across known interventions, also biomarkers at the bulk, single cell level, causal, multi-tissue, also different omics level like epigenetic, transcriptomic, and proteomic, and also mentioned about biomarkers of aging consortium. I invite you all to the conference that will be Friday and Saturday this week at the Harvard Medical School. Also, I mentioned about reversible changes in biological age upon severe stress or some other forms of adversity. Again, I mentioned 4 cases, but then we have more now in the lab. Also, we discussed rejuvenation, and I told you about 2 examples, heterochronic parabiosis and also rejuvenation during early development that illustrates this paradigm on when aging begins. We think it begins around the stage of gastrulation in mammals or in vertebrates in general. And finally, I discussed organ-specific aging, which resulted from the studies based on proteomic profiles, models, chronological mortality models, association with diseases and diets, and also this concept of considering disease as a localized, non-autonomous aging process. Ultimately, our studies focus in the lab on understanding and targeting aging, longevity, and rejuvenation. And we use a variety of approaches, you can see, and one can say there's progress. There's a progress. On the other hand, I may say at the very beginning of the road, because we really don't understand still fully very, very basic questions in the field, like starting from the nature of aging and the nature of rejuvenation, how to quantify it. But to me, there is some progress, at least in this direction. But there's a way to go to develop better biomarkers and quantify and use them in various ways. Finally, this is the current lab. Current members of the lab, but also many previous lab members contributed. I mentioned some of the people whose studies I showed you, and also we have many collaborators and also funding sources. So with this, I would like to thank you for listening, and I look forward to answer some questions. Thank you.
Thank you so much, Vadim, for a terrific, characteristically very thought-provoking seminar, as always. Just a reminder to everybody to use the Q&A for questions, and I will read them out. The first one came through the chat from Harry Dawson. Regarding the stress-related clock, are there correlations between whether the species produces cortisol or corticosterone?
No, we don't know that. So with the stress, with the resolution of current clocks, we observed the effects under conditions of severe stress, like mild stress typically doesn't show any effect. And at the molecular level, which conditions represent a severe stress condition, which are not, still needs defining. It's kind of the broad umbrella of severe stress to encompass multiple kind of states of severe adversity that we observe. But I think there are more studies needed on that.
Next question is from Larissa Constantinou. Thank you for the fascinating talk. Has anybody tried to use changes in extracellular proteins as biomarkers of aging, such as glycosylated collagen?
There is a glycan age. This Gordon Lauk from Croatia, he developed the glycan age, which quite accurately quantifies biological age. I think it's not as good as biomarkers based on the proteome or the epigenome or transcriptome, but can be used as also as an additional biomarker.
Thank you. Michael Lustgarten says, hi, Vadim, and thank you for your many contributions to the field.
Thank you.
In terms of parabiosis in old mice extending lifespan, do you have any thoughts on the recent preprint from the Kennedy lab, which suggests that median lifespans more than 900 days should be considered as significant? Within that context, median lifespan of the old young parabiotic mice was only 875 days, 125 weeks.
You're right. So sometimes mice live slightly different lives in different labs under different kind of environmental conditions. These studies should be used as a model, right? That we study. We try to use the best controls that we have that treated in the same way under the same environmental conditions, same facility, same diet, and so on. Treatment by the same individual lab technician. But the studies are imperfect, of course. And it may be that under some conditions that we had, mice for some reason lived a little bit shorter. And for example, if this is repeated with under conditions when mice become a little bit longer lived, the effect might be increased or decreased. That's perfectly possible. I already mentioned that we observe a smaller longevity effect than would be expected based on the observations of rejuvenation early in life. So our interpretation is that the effect diminishes over time.
Our next question is from Harry Ecker. Hi, Vadim. Thank you for the fantastic data and talk. Could you share insights on the mechanisms of heterochronic parabiosis leading to rejuvenation? Also, are these effects observed across all organs?
We observed the effects across all organs that we tested. We think it's a very robust procedure, but mechanistically, we don't understand exactly how this happens. We're currently studying this because the effect appears to be very complex. It's not simply the transfer of longevity signals or youthful factors from younger mice to older mice, and not simply like dilution of damage by connecting the blood. Because in addition to exchange of the blood, older mice, they have access to the younger organs. They're able to detoxify, to activate the detoxification process to export damage through these organs. So the effect seems to be complex, multifactorial, and not really well defined yet. So in that sense, I'm a little bit skeptical of the simple procedures that I use to recapitulate. For example, like injecting young blood, I just don't see how it's incompatible. So for example, when we've done this parabiosis, we've done it for 3 months, but we also have done for 1 month. And 1 month effect is much weaker than the 3 month in terms of the rejuvenation. So, and of course, even 1 month, it's a very extended because when mice live 3 years, like 3 months is like 10% of their life. And even then we observe like the effect which can be diminished by exposing them only for 1 month. And when we do like injection of young plant, to me, it should be very minimal effect. And even to the extent that procedures like plasmapheresis and other similar procedures, I think they expected to have much milder effect compared to heterochronic parabiosis.
Next question from Jillian Ritter. What do you think calorie restriction reduces, or why do you think calorie restriction reduces aging in your models? And have you explored the role of apelin in aging?
Well, mechanistically, I mean, we don't particularly study calorie restriction, right? In terms of the pathways, we just know that in our model, at least at the omics level that we study, calorie restriction is a part of the group of interventions that very similarly change the mouse. These interventions include, for example, FGF21 overexpression, growth hormone receptor deficiency, some genetic model, some dwarf mice like Snell mice, Ames mice, also includes fasting, and also includes every other day feeding, those kind of things, but quite different from rapamycin, for example. But in terms of the individual interventions like apelin, we don't really study this. So we try to approach aging from the systemic perspective. And once we decompose this extensively to individual molecules, then we often lose the signal. So we try to describe aging like as a system, like the entire system, rather than kind of individual genes and components.
Saidas says, excellent talk. Do you know if the association between organ-specific aging and diet varies with population subtypes, and is it replicable?
Oh, it may be. I mean, we don't know. I mean, of course, it would be expected that it would be the case. But what I presented is really new. I mean, this was done this year. It's still unpublished. And we really haven't studied this at the level of populations. Maybe in the future with better data sets, it may be possible.
From Larry Parnell, have you observed any age-related changes in the crosstalk between organs?
Age-related changes in the crosstalk among organs. We have several projects I haven't presented today, but we have also projects on transplantation. We study like when we place an organ of different biological age in the animal. It kind of relates to the question of partial reprogramming when many labs and companies, they target particular organ with Yamanaka factors and trying to rejuvenate them. So the question we ask, if we assume that we fully rejuvenate an organ, we just simply replace, like for example, we take the young kidney and place in the old mouse. Let's say it's equivalent to full reprogramming, to rejuvenation of the organ. How would affect the aging of that organism? How would it affect aging of that particular organ? So we have several models like that currently in the lab, and we actively studied this, but there is no clear understanding yet, because I would say preliminary that the assimilation of age of the organism is obvious. So for example, if the organism has a particular biological age and we place organ of a widely different biological age, that organ will rapidly move to the age state of the organism. But it's unequal across different systems. And again, so it just needs to be studied more for more robust conclusions.
Thanks, Vadim. We have 3 more questions that I'm going to ask if some of you have to head off to other meetings. Thank you for coming. So the next question is from Sheldon Rowan. How do you view cancer in relationship to aging in contrast to other organ-related diseases? There didn't seem to be much correlation.
And actually, it's a good point. So in the slide where I showed that diseased organs, they show increased age, there was one outlier, and that outlier is cancer, interestingly. So cancer, it seems like based on our preliminary studies, cancer may be older or maybe younger. It's really various. There's a lot of heterogeneity, and somehow it's not very well studied, and we certainly have not focused yet on that topic extensively enough to present. And for some reason, it's also largely absent from the literature, which is quite surprising. It's a really interesting topic to explore.
Another question from Saidas: does maternal age influence zygote age? And if so, what might be the influence?
We predict that it would, but there's no hard data on that. We actually have a small project to test this effect in the lab currently, but we don't have the data yet. But certainly there would be an expectation that not just a zygote, but even the blastocysts, they would be expected to be older from older parents.
And then one last question from Thomas Biederer. Thank you for an outstanding seminar. It would be great to learn your thoughts on aging as a risk factor for neurodegeneration from humans to mouse models. What are, in your opinion, the most informative approaches to understand this?
We do not particularly focus on neurodegeneration. I mean, we just focus broadly on aging. So brain, of course, is an important organ, so influences aging of other organs very clearly. But I don't have any smart things to say here. Unfortunately, it's just not our focus.
For those of you who are going to be able to attend the Biomarkers of Aging Symposium, maybe you can push Vadim on this question and others. I found that given time, he always has very interesting thoughts about any question that comes his way. Thank you again so much for a terrific seminar, Vadim, and thank you to everyone for being able to show up, and hope to see you soon.
Yeah, thank you very much. Thank you. Okay, bye.